Welcome to this class, BTH 201, Introduction to Biotechnology. These are the main outcomes which you already have and have gone through, and these are the course aim to enable you to have a basic understanding of biotechnology and genetic principles and methods in the various aspects of biotechnology. And these are objectives to give you a historical perspective of biotechnology from traditional to modern, identify the various disciplines involved in biotechnology, identify the various tools used in biotechnology, illustrate and describe the techniques of cloning, discuss the scientific basis of biotechnology, describe the central dogma of molecular biology and its importance in biotechnology, understand the use of microorganisms in biotechnology, explain the use of microorganisms in production of biofuels, and so on. Now, look at the first model. What is biotechnology? We discussed this in the previous lesson, and you're given some materials to go through. Bill Gates once said that two technologies that will shape the next century are biotechnology and information technology. Biotechnology is not so new. It began as far back as 1919 when Carl Ericke wrote a book called Biotechnology of Meat, Fat, and Milk Production in an agricultural large-scale farm. For him, he indicated that the processed biological materials could be biologically upgraded into socially useful products. That's what was termed biotechnology. But looking at the definition given by United Nations Convention on Biological Diversity, you can define biotechnology as any technological application that uses biological systems, living organisms, or derivatives thereof to make or modify products or processes for specific use. Now, when you study biotechnology, there are several disciplines that are involved, as you will see shortly. You're going to study genetics, biochemistry, cell biology, statistics, a bit of chemical engineering, talk about process biotechnology, plant and animal breeding, food science, among others. There are basically two types of biotechnology. You have the traditional biotechnology that has been on for centuries, even before the time of Christ. In terms of plants and animals, talk about domestication. You did a bit of agriculture, so you notice that you have the rearing of crops and animals. The good seeds of crops are kept by the farmers and conserved and planted in next year. Same thing, animals were crossed one with another to get better products from them. Wild animals were also controlled to produce milk or meat. In terms of pest control, using things like ash and neem extract from the neem tree have been proven to be able to control insects, to control ant attack by plant ants and termites on banana and yam seedlings which you plant. So these are traditional methods of biotechnology. Also in the food industry, the traditional forms of biotechnology include production of vinegar, cheese, yogurt, your palm wine, different types of alcoholic wine, beer, all as a result of fermentation by microorganisms. You also have fermentation of cassava, your maize, your flour enhancers. So you can see the traditional biotechnology has been on for a given period of time. It's not something that is new. If we look at the traditional biotechnology, I talk about under food, you will see that certain microorganisms have been used in the past. You have your yeast, Saccharomyces cerevisiae, and the main product it gives is alcohol in case of beer. In case of bread production, it produces carbon dioxide, which enables the dough used in making bread to rise. Also, you have yogurt, either produced by Lactobacillus streptococcus, and this produces lactic acid. You will notice there are different types of yogurt in circulation now, and each has its own distinct taste, and this is linked to the particular strain of microorganism that is used in producing the lactic acid. One thing about this conventional method of biotechnology or traditional plant breeding is that number one, you can only make crosses within organisms of the same species. You cannot transfer genes from maize to wheat or from cowpea to banana. No. It's always cowpea, maize to maize. So it has to be organisms of the same species. Secondly, apart from the gene of interest that is transferred, there are other genes which can also be transferred, which are not under the control of the plant breeder. It may be detrimental or it may be useful. Thirdly, it takes a long time to get the improved variety, sometimes 8 to 12 generations. Because you have to do a lot of backcrossing to make sure that the gene is integrated into all cells of the plant. And lastly, it's a natural process. We are crossing the crops naturally. Of course, this came about from the work of Gregor Mendel, who is regarded as the father of genetics. But these days, plant breeders make use of molecular breeding method or marker-assisted selection to circumvent the limitations of conventional plant breeding. Another thing about conventional plant breeding is that you have to wait for the plant to grow to maturity before you select which plant to use as a parent in your crossing. But with marker-assisted selection, the limitations have been circumvented. You can plant different, let's assume you have 50 or 100 genotype of a particular crop, let's say cowpea. I'm trying to look for some useful genes in some of them. You know which ones you are going to use for crossing as parents. And if you have knowledge of that gene, you can design a molecular marker that targets that gene and then extract the DNA of the plant at the 2 week stage when the plant is still on 2 weeks old and run the PCR with the DNA and the molecular marker. If that plant has that gene, the molecular marker will easily detect it. As you can see here, it will give you bands. Here you have the phenotype on the right and the DNA bands on the left. If you look at the middle, the bands are thicker, which shows that these ones have that particular gene that is being looked for by the. Also, the conventional method has to do with selective breeding for improved food production, breeding for desirable crop traits, and to alter disease vulnerability. Because you don't want your crops to be attacked by pests and diseases. Also here you can see the diagram of the ancestor of the modern the tomato and also compare teosinte with the medical. Like I said, biotechnology, traditional biotechnology has been on for ages. Domestication of plants started around 8,000 years before the birth of Christ. And you go on, you can say by 3000 BC, you have bread production. The Egyptians were very good at that. Cheese production, plant breeding, the work of Gregor Mendel, the father of genetics, who came up with the laws of inheritance, independent assortment, and the law of segregation. They have discovery of the BT gene, which is from an organism called Agrobacterium tumefaciens, which is natural plant genetic engineer. Now, moving into the era of modern biotechnology, things began to change. The Cartagena Protocol on Biosafety defined modern biotechnology as the application of in vitro nucleic acid techniques or fusion of cells beyond the taxonomic family that overcome natural, physiological, reproductive, or recombination barriers. Take note of this definition is different from the general definition given for biotechnology based on United Nations Convention. We're talking about the in vitro nucleic acid techniques. We're talking about DNA, RNA. In vitro means outside the living system, and a test tube, the append tube. Fusion of cells beyond the taxonomic family. This talks about protoplast fission. A protoplast is a plant cell devoid of the cell. So fusing protoplasts of two different species together can give you a hybrid. And all this overcomes natural, physiological, reproductive, and recognition barriers. Why? Because you cannot transfer genes across species, which naturally is not possible. So this is an artificial method of enhancing growth of crops and animals and introducing useful genes into them. It is very unique simply because it permits scientists to precisely introduce a desired trait by inserting only those genes into plants and animals, because you know the function of the gene and you know the metabolic pathway or biochemical pathway the gene has in the system. So it is more precise than the conventional method. So it is used to produce transgenic animals as well as transgenic plants. This also gives you a brief flow of the beginning of modern biotechnology. The plant tissue culture is also traditional biotechnology, but now you have temporary immersion bioreactor, which is much more advanced. You have the first recombinant DNA drug produced in 1982, human insulin. In the past, insulin was obtained from the gut of pigs or cow, as porcine or bovine insulin. But definitely humans reacted to it because it's not coming from the human gene. So there are a lot of allergic reactions and some died from it. But with the advent of recombinant DNA technology, especially with the discovery of restriction enzymes or endonucleases, which make specific internal cuts on DNA, recognizing palindromic sequences. These restriction enzymes can cut the DNA of different species, a micro plant, and give you similar um, fragments with sticky ends. So with this knowledge, scientists have cloned genes from different species into a circular DNA molecule called a plasmid, seed it up with DNA ligase, and transfer it back into the bacteria. And as the bacteria multiplies, it begins to produce products of that gene. So in 1982, human insulin called homolin was produced. Since then, a lot of biotech crops were produced. Mind you, you notice that as we move from traditional biotechnology to modern biotechnology, it becomes much more expensive. Microbial fermentation, biological nitrogen fixation, plant tissue controls are traditional methods. Instead of advanced embryo transfer, like Dolly the sheep that was produced in 1997, monoclonal antibodies, and then with recombinant genetic, we talk about genetic engineering of microbes, animals, and plants. So we've seen a brief history of biotechnology. You can read more about it. There's a lot of information on the internet and also in the library in textbooks. But we also have spectrum of biotechnology. This divides biotechnology using different colors based on the area. For example, when you have red biotechnology, it talks about medical aspect of biotechnology. But blood, as you know, is red in color. The medical procedures uses microorganisms for production of novel drugs, biopharmaceuticals, or replacement of tissues with healthy ones. For example, talk about gene therapy, organ transplantation, and all that, medical biotechnology. When we talk about green biotechnology, it also refers to agricultural biotechnology because it has to do more with plants. It applies agriculture and involves processes such as the development of pest-resistant grains and the accelerated evolution of disease-resistant animals. It focuses on agriculture. They are interested in yield of crops and better growth of animals, better milk and all that. In the case of blue biotechnology refers to more to the environment, processes in the marine and aquatic environment, so they are controlling the proliferation of noxious waterborne organisms or enhancing the lives of aquatic organisms, having fishes that are temperature tolerant or that grow at particular rate are resistant to diseases. Okay. In the case of white biotechnology, it involves industrial processes. Especially we talk about production of clean fuel as alternative sources of fuel apart from hydrocarbons. So we talk about your biofuel and other industrial products produced by biotechnology. So in brief, this gives you an idea of how biotechnology is classified as medical, having to do with human health, and pharmaceutical industry. The green has to do with agro-food. The blue has to do with aquaculture. The white has to do with industrial applications. You also have biodegradable plastics as well coming into this line, as well as textiles. For example, you have your BT cotton. And here it has to do with environmental control and regulation, talking about cleaning up of polluted environments using biotechnology. So let's look at the different spectrum of biotechnology in detail. When we look at the red biotechnology, they are producing vaccines and antibiotics. One thing that came out during this recent COVID-19 is the issue of vaccine production. For the first time, an mRNA vaccine was produced. That's the Moderna vaccine against the coronavirus. We talk about the development of new drugs, molecular diagnostic techniques. It's becoming easier to detect individuals that had the COVID virus. Talk about cell therapy or gene therapy, replacing faulty cells with healthy cells, and other therapeutic antibodies being produced. In case of agriculture, which has much more focus, we talk about creating new plant of agricultural interest, pest and disease resistant crops. You have your BT cotton, BT maize, and now you have the poor bar resistant cowpea that was produced in Nigeria at the Institute of Agricultural Research and Training, Samaru Zaria, all against lepidoptera insects that affect the crop. Of course, this gene taken from the bacterium Bacillus thuringiensis has no effect on man, but only affects the GIT of the insects when trying to feed on the crops. Now we're in the era of climate change, so scientists are working to get climate resilient crops, and this can only be made possible with the use of modern biotechnology. But it doesn't mean that the conventional method are not used. After the biotech crop has been produced, they need to integrate that gene into the commercial varieties of the farmer. Many products and applications from blue biotechnology as object of study and research. There are a lot of organisms, living organisms in the aquatic environment, and some of these organisms have useful genes that produce secondary metabolites that are useful to man. Studies are still going on this area. Under our white biotechnology, talk about chemical production using microorganisms to produce ethanol, biodiesel, to design and production of new materials for daily use, biodegradable plastics. You can notice that here in Nigeria, the environment is polluted with a lot of plastics, but in developed countries, biotechnology has been used to produce biodegradable plastics. When we throw them into the environment, they decompose and cause less damage to the environment. Also, the development of new sustainable energy sources, such as the biofuel. Now, let's move to the second unit of this course, talking about the historical development of biotechnology in Nigeria. It's suffice to know that biotechnology began in Africa as far back as 1997 in South Africa. So South Africa is the first country that began biotechnology in Africa. In 2001, Nigerians had interest and began to plan for the biotechnology development. And by 2001, we had the National Biotechnology Development Agency, NABDA, established in Abuja. By 2003, we have centers of excellence established in the 6 geopolitical zones. One at UI in west of Ibadan, we have one at the University of Nigeria Nsukka in the east, we have Ahmadu Bello, Zaire, and the other 3 regions, each having a university. By 2005, we had the first biosafety review of applications for confined feed trials of transgenic cassava, really the first crop to have been genetically modified in Nigeria. So that project continued till 2009, where the feed trial for biofortification cassava was carried out with beta carotene. Because if you look at the South Nigeria, you have the yellow gari there because you had palm oil to the gari to give vitamin A to it. That's an artificial method. But now with modern biotechnology, also conventional biotechnology, it can also be used. The plant begins to produce the beta carotene. When you cut the tuber of the cassava, it is yellow in color because it's producing the beta carotene. By 2011, we have 3 adapted Nigerian sorghum varieties. The confined feed trial was completed. Why confined feed trial? When we are carrying out biotechnology, you have to ensure that there is no harm done to the environment. And this is done by planting those GM crops in an environment, isolated environment, far away from all other crops to prevent introgression of genes into other crops. But biotechnology did not get the boost it required until 2015, when the former president, Goodluck Jonathan, signed the biosafety bill, which made Nigeria eligible to join other countries in genetic modification of crops to alleviate poverty and malnutrition and enhance food security. By 2018, Nigeria now produced 2 homegrown cotton varieties, which is giving farmer a lot of yield now. And by 2019, the pod borer resistant cowpea to store the millions of legume pod borer, Maruca vitrata, was completed. Now, other African countries, especially Ghana, are now running to Nigeria to find out how they can make use of this technology and enhance cowpea production in their country. So, looking at Nigeria, there are different regulatory bodies for biotechnology safety in Nigeria. You have the Federal Ministry of Environment, you have the National Biotechnology Development Agency, which ensures that application of modern biotechnology and the handling and use of genetically modified organisms are not harmful to the conservation and sustainable utilization of biodiversity, taking into account risk to the health of Nigerians. In summary, they ensure biosafety and biosecurity in Nigeria. Also, the National Biotechnology Management Agency, which have different sections: agricultural, medical, environmental, bio-entrepreneurs, and extension services in biotechnology. Then when it comes to food and drug administration, NAFDAC also have a section on quality control to ensure that whatever is being displayed as biotech crops have no detrimental effect to man. At the international level, you have United States regulatory bodies such as the United States Department of Agriculture, Environmental Protection Agency, Food and Drug Administration, Animal and Plant Health Inspection Service. This focuses more on agriculture, but now you have other bodies that control the area of medical and pharmaceutical technology. Like I said in the beginning of the course, there are several disciplines involved in biotechnology. If you look at the tree, you see on the slide here, you can look at the roots, which are the disciplines involved in biotechnology. As a biotechnology student, you will need to study computer science. Take a course in computer science at 100 level, which you have already done. At 100 level now, you're going to study genetics, introduction to microbiology, biochemistry, and you're going to do statistics. So you want to analyze your data, just as Gregor Mendel did. And on top of the tree, you can see different applications of biotechnology with the help of the various disciplines. There are genetic engineering through molecular biology and analysis through bioinformatics, environmental and aquatic biotechnology, agricultural biotechnology, pharmaceutical and medical biotechnology, also the regulatory bodies, and so on. So biotechnology is a very interesting course which you are studying now and you should take it seriously so you learn more about it. Lastly, for today's class, we're going to look at the tools used in biotechnology. We talk about gene cloning. It means identifying and isolating specific DNA fragments and introducing them into a self-replicating genetic material called a plasmid. These plasmids are found in bacteria, only prokaryotes, and is different from the chromosome of the bacteria itself. So the bacteria multiplies, the DNA of the circular DNA plasmid also multiplies. So scientists have not made use of recombinant technology using restriction enzymes to cut the gene of interest and using restriction enzyme to cut that of a specific plasmid, given sticky ends, and then mixing the fragments with the plasmids and sealing off the gap with DNA ligase, giving you a recombinant plasmid. This recombinant plasmid is then transformed back into the bacteria where it multiplies and produces a gene product, and then finally that gene product is purified. In the case of tissue culture, it is a technique that involves culturing plant and animal cells in a laboratory under aseptic conditions. There are different techniques used in tissue culture, be it plant or animals. In the case of microbial culture, a method of multiplying microbial organisms. In molecular plant breeding, I've already explained this area earlier in the course of this lecture. A technique using DNA markers that are highly linked to specific traits which the plant breeders look out for, all in a bid to assist in selection scheme for a particular breeding objective. You don't need to wait for the crops to grow, plant to go to maturity before you determine which plant to use as a parent. Young plants are 2 weeks old, the DNA can be extracted from the leaf, and then select molecular markers targeting specific traits can be used to identify those plants that have those traits. So DNA marker technology involves using DNA fragments and so setting desired trait and isolation using DNA or genetic markers is a follow-up to molecular plant breeding. So we have different molecular markers that we use. But now we can have a high throughput method to our diversity array technology and so on. When we talk about genetic analysis, has to do with studying how traits and genes for traits are passed from generation to generation, and how genes and environment interact to result in traits. Because definitely there's an interaction between the genotype of an individual and the phenotype. For example, you have two maize, like you plant one on loamy soil and the other on clay soil. They will not give you the same yield because the environment affects the expression of the genes. When you look at genetic engineering, it has to do with manipulation, introduction, and expression of specific genes or DNA in a target organisms. This is also sometimes called genetically modified organisms, because foreign genes have been inserted into them. Sometimes it's called modern biotechnology. When you hear modern biotechnology, gene technology, genetic engineering, recombinant technology, they all mean the same thing. Then you also have genome editing, a new technology that has opened up the possibility of directly targeting and modifying genomic sequences in almost all eukaryotic cells using different methods. That is zinc finger nuclease, the transcription activator-like effector nuclease, but the most commonly used one is the CRISPR-Cas9 called clustered regular interspaced short palindromic repeat associated 9 nuclease. This again is obtained from bacteria. Scientists have learned how to use this to snip off faulty genes. So it's different from genetic engineering. We are always adding genes. Well, this one has to do with removal of genes that are not useful, and it has less regulation than that of the genetic engineering method. The bioinformatics and the scientific field of science that develops method and software tools for understanding biological data. There are a lot of omic technology now. We talk about genomics, proteomics, transcriptomics, and huge data is developed. So using computer software to explain biological processes is what bioinformatics is all about. It enables scientists to store, organize, annotate meanings, find the function of the genes, visualize, code, and interpret this data. When we talk about genomics, it has to do with the study of the complete set of DNA present in an individual, the sequences, that means order in which the four nucleotide bases, adenine, thymine, guanine, cytosine, are arranged. Proteomic means the analysis of the complete set of proteins or protons in an organism. You know very well that from experimental molecular biology, the DNA gives rise to RNA and RNA gives rise to protein. Therefore, gene expression has two stages: transcription, information from DNA copied to RNA, and translation, information from RNA translated into protein. Also have diagnostics, detection of pathogens in plants, animals, and humans. Then lastly, vaccine technology. Like I did say, this vaccine is made from killed or weakened forms of microbes. But now with the COVID-19, we are now looking at using modern biotechnology methods to produce vaccines. For example, viruses are made up of protein coat. So the messenger RNA that gives rise to that protein coat is now being used to develop immunity against the virus itself. So the immune system is stimulated to recognize the agent as a threat and destroy it. And then all these microorganisms that it later encounters. That's how we build immunity against viruses. And that's how the war against coronavirus was achieved. We stop here in today's class. We continue from there in the next class. Make sure you go through the materials that you have been given, read more about it, and you're going to be given an assignment to keep you busy.